Now in this class we are going to talk on next part of the DNA sequencing. At first, before coming on the today's topic, we want to analyze the bands of the previous class again, because some students communicated me that they have no proper understanding for the analysis of the data. So again I am revising only the data analysis part or the analysis of the banding pattern in the last class. In last class, suppose 4 reactions I was discussed on the 4 reactions. First, dimethyl sulfate plus piperidine. It is responsible for separation of guanine, and second reaction, dimethyl sulfate plus piperidine plus formic acid. It produces guanine plus adenine. Third one, hydrogen plus piperidine. Produces cytosine plus thymine. And fourth one, hydrogen plus piperidine plus alkali, only cytosine. In this way, if we analyze the only one, suppose simply it is the first reaction, it is the second reaction, third reaction, and fourth reaction. In the first reaction, here the guanine band is present. In the second reaction, because the guanine is present, that means here also a band is present due to the presence of guanine. But if the band is present only at the second point, but not at the first reaction, in the first reaction, it represents A and it represents G. Likely in the third reaction, it is responsible for the here due to the presence of the cytosine, and if you come here in the hydrogen 4, it is also present a band here in the fourth reaction also. But if you observe a single reaction in the third one, but not at the fourth reaction, here it represents the cytosine and it represents the thymus. It is the basic binding pattern. That means if in the pyrimidine line 1 and 2 represents the purine line and 3 and 4 represents pyrimidine line. In the purine line, if two bands are present, in both first and second, it represents G. But if one band is only represent, in the second line, it represents A, adenine, adenylic deoxyribonucleotide. Likely in the third band, it represents the C if both bands are present in the third and fourth line, and T if the band is appeared only on the third line. Now, I suppose that it provides a clear-cut concept for the analysis of banding pattern as the Maxwell-Gilbert method. Now, we come on a band. Suppose a autoradiogram represents it is the first reaction, second reaction, third reaction, and fourth reaction. In first reaction, a band is present in the first reaction and in second reaction. Here again in the first reaction and in second reaction. Now it is present in the third reaction and fourth reaction. In the third reaction only. Here in the second reaction only. Now a band is present here in the like this. Again a band present here and here. Like this. If you observe a band like this, then it is clear that the band represents the G. Here the band again represents G. Here it is C. Here it represents T, A, G, T, C, G, A, C, T, and G and A. In this way, we can analyze the banding nature and corresponding DNA sequence, because we analyze the DNA sequence by spotted structures of the band dispersed throughout the electrophoretic field. It is referred as wandering spot analysis. But the method is time consuming and sequence limiting. In addition with these two problems, the some chemicals are harmful to the man. Hence, some alternate method, and it is better to say here that an alternate method was developed by Sanger in 1975, and it is referred as sequence termination method. Or chain termination method. Chain termination method. The basic principle of the chain termination method is very simple. We all know that the DNA is basically formed by the nucleotide and here the one nucleotide and here it is another nucleotide. This one at first the phosphoric acid binds to the fibracid. Here the phosphoric acid also binds to the fibracid. And after the polymerization, after the formation of the nucleotide, these nucleotides are connected by the 3' and 5'. 3' and 5'. That means the polymerization is direction of the polymerization is from 5 to 3 direction. It is the basic pattern of the polymerization. That means For the polymerization, this part is most important. That is the deoxyribonucleic acid. Sanger, the principle of the Sanger is based on the ribo nature of the deoxyribonucleic acid. That means the sugar here that is the ribonucleic acid. Observed in the ribose. Two dash deoxyribonucleic acid. 2-deoxyribose observed in the deoxyribonucleic acid or DNA. Here, a third pattern, that is the H, 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 OH, CH2OH, NH. That is 2-3-dideoxy ribose. And Sanger used this particular nucleic acid in the polymerization, and the use of 2-3-dideoxyribose is responsible for the chain termination of the polymerizing sequence of DNA, and it provides the basis for DNA sequencing. Here, again, we use 4 reactions. In reaction 1, reaction 2, reaction 3, and reaction 4. We commonly observe that during the polymerization, D2-deoxyribonucleotide triphosphate of the all basic components are required. And here, in the first D, ATP, D, CTP, D, GTP, D, TTP. We use all these building blocks in all 4 reactions. Again here, DATP, DCTP, DGTP, and DTTP. Likely, DATP, DCTP, DGTP, DTTP. Again in fourth reaction, DATP, DCTP, DGTP, DTTP plus DNA polymerase, Taq DNA polymerase, because Taq DNA polymerase is stable, heat stable. Here again we use the Taq polymerase, Taq DNA polymerase. Here again we use Taq DNA polymerase. Here again we use Taq DNA polymerase plus primer, plus primer, plus primer, plus primer here, plus DD, that means the di 2 dash 3 dash di deoxy adenosine triphosphate. It is the block which is especially used by the Sanger in 1975 for the analysis of the DNA sequence by the chain termination method. Again 2 dash 3 dash DDTTP. Again 2 dash 3 dash DDGTP. 2 dash 3 dash DDGTP. And for all these analysis, we use the desired single stranded DNA. Desired single stranded DNA as a template. And the desired DNA single stranded as a template, if we use in all these 4 reactions in the separate chamber, the polymerization is initiated and developed, but the polymerization will be stopped when the ATP is present by the process of the chain termination. And after the completion of the reactions, the products are the reaction products are placed in polyacryl amide urea gel electrophoresis for separation. The bands are separated by the polyacrylamide urea gel electrophoresis. And after the separation, the bands are studied either in UV light or by auto radiography or by auto radiography. It is the basic method, and when suppose a model for the study and analysis, and suppose a DNA segment is reactions are placed in the electrophoresis. The basic electrophoretic principles are same. That is, the lighter sequence has more faster movement, and support is the reaction 1, it is the reaction 2, it is the reaction 3, it is the reaction 4. Here the band is, we observe in this sequence, we observe the band sequence like this. And it is a direct method. Here we have no any comparison and the analysis. Here the first band we use the, here in the first case, DDATP. That means all the bands observed in this lane represent the A. In this second we use the DDTTP. That means all the bands represents here that represented the T. Here the DDCTP. That means all the bands represented here is actually represent C. And DDGTP. Sorry. That means here all the bands we represent here represent the G. That means here the sequence is at first T. That means the sequence is T. Second one A. That means TA. Third one here TAC. That means TAC. This one, fourth one, that is G. GCAT. Fifth one, T. GCAT. It is the first band, it is the second band, it is the third band, fourth, fifth, sixth, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. And now you analyze, first is the T. Second is A, that means T and A. T and A. Third one is C. C, T, A. Fourth one G. G, C, T, A. Fifth one T. T, G, C, T, A. Sixth one A. A, T, G, C, T, A. Seventh one G. G, A, T, G, C, T, get one C. C-G-A-T-G-C-T-A. Likely, we come here and finally we are in a position to analyze all the DNA sequences. Here, in the chain termination process, of course it is uh, better. It is better than the Maxim Gilbert method, but it has also some limitations. First limitation, that is, it is also the time consuming, and for the analysis, we depends on the indirect method. It is not direct method, that is the banding pattern. Of course, it is superior than the Maxim Gilbert method. So further, we developed it by the use of fluorescent dye, fluorescent markers we used for the in addition with the method of Sanger is developed further by the use of dye terminator sequencing. Of course, it is the method based on the principle of Sanger, but it includes a terminator tagged with the fluorescent dye. Dye with different wavelength. With a different wavelength, and due to the presence of different wavelength of the chain terminator, we can in a position to process all the DNA that is not in the four different reactions, but in a single reaction. In a single reaction. And after the processing in a single reaction, because each terminator has a different wavelength blocker, and each wavelength is characterized by a different color emitted by the emitted from the blocking side. We in a position to analyze exactly the DNA sequence by a chromatogram. By a chromatogram, and it is the now a machine is developed known as DNA sequencer. DNA sequencer. Now we use a automated machine, that is the DNA sequencer, which is responsible for the sequencing as well as production of the chromatogram, and chromatogram is also analyzed by the by the commercial or non-commercial available software. For example, suppose a DNA sequence has A C G A T C A T A C C G. It has the three sequence, that is fibrous the three test. And if we analyze in the DNA sequencer, it is clearly represented here that it has the separate wavelength. A blocker, a DNA blocker, has the separate wavelength, and suppose the A has the blocker of the yellow dye. C has red. G green. And T blue. And if we do DNA sequence, it represents the different wavelengths, and that wavelength emitted light with the different intensity, because here the intensity is magnitude of the intensity in light, intensity of the light is increased by the number. Here, for suppose here it is the 4. Here the in sequence it is the 4. That means here the wave of A. It is the wave of A. It is the wave of C. It is the wave of G. Here 1, 2, 3, 4. That means here 4 times wavelength is for the A. Again T, like this. Again C. A. T. A. Here again double C. That is like this. And this one that is the chromatogram. This represents chromatogram. And by this process, we analyze the DNA sequence in a single reaction. And after the development of the automated and use of the fluorescent dye, automation machine, and the techniques, the process of the DNA sequencing is developed. But the DNA sequencing by the process of the chromatogram has also some limitations, because if we use up to the 14 or 15, earlier 14 to 15 nucleotides, the wave or chromatogram is not perfect, and likely when we move from the roughly 780 nucleotides and above, it has also some limitations. And now today we discussed on the different methods of the DNA sequencing, and in next class we will discuss on the pyro sequencing. Thank you.